Hi, Dr. Crowder here, and I'm making this video to go over a brief summary of the Lac Operon and how you can analyze different Lac Operon mutants as far as expression of the operon genes. But before we go into the operon, I want to take a few steps back and just talk about why typically in genetics classes you are introduced to the Lac Operon. And really, the Lac Operon is a model system for how prokaryotes, how bacteria regulate gene expression. And so it's the most well understood example of how prokaryotes can regulate gene expression, largely due to the work of Francois Jacob and Jacques Monod in the late 50s and early 60s to kind of elucidate this system. So let's look at E. coli, which was the model system that Jacob and Monod were working on. And E. coli bacteria will take up different sugars in their environment and use those as a source for carbon and energy. So I'm showing you a couple sugars here: glucose, galactose, lactose, and xylose, all of which E. coli are able to use as a carbon source and for energy. However, E. coli really only want to turn on expression of the genes involved in metabolizing each of these different carbon sources whenever that is the carbon source available to them. So for example, if glucose is available to E. coli, it's actually the preferred carbon source, you want to turn on, or not you, but E. coli would want to turn on all of the genes, so transcribe and translate all of the genes that encode enzymes necessary to metabolize glucose for its carbon and energy, and turn off genes that encodes enzymes necessary to metabolize galactose, lactose, or xylose because it's not metabolizing those compounds. And so in order to do this, bacteria have to, one, sense the environment to see what type of carbon source is available to them, and then be able to quickly turn off or turn on genes that are necessary for metabolizing the carbon source available. So with respect to the Lac Operon, if lactose is available, the presence of lactose as a carbon source, bacteria have a mechanism to be able to sense that lactose is available, and in response turn on or express genes encoding enzymes that function in lactose metabolism. And so this is done through the Lac Operon, which again is an example that is typically used to demonstrate how bacteria or prokaryotes regulate gene expression in response to their environment. And again, it's work done by Jacob and Monod in the late 50s and early 60s. So before we go into analyzing mutants, let's just dissect and break down the different components of the Lac Operon. So I'm showing you here a representation of a portion of a bacterial chromosome with some of the genes, LAC-Z, LAC-Y, and LAC-A, that encode enzymes responsible for metabolizing lactose. So let's just consider a case where transcription and translation is occurring and all of the genes shown here are being expressed. So we have RNA polymerase shown in red. RNA polymerase will bind to the promoter, which for the Lac Operon, the promoter is shown here in green. And RNA polymerase will bind and transcribe the genes in a polycistronic mRNA as shown here. Now, to point out, we have LAC-Z, LAC-Y, and LAC-A, which are termed the structural genes of the Lac Operon. These are the genes that encode enzymes responsible for metabolizing lactose, and they are all under the same transcriptional control. So they are all under the same promoter and an operator sequence, which we'll talk about more. We also need to point out the LAC-I gene. This gene is not specifically on the operon or part of the operon, but it's important because it encodes the repressor protein. So the LAC-I gene is under its own regulatory control. but it's important, again, because the repressor protein is involved in controlling the Lac Operon. So if RNA polymerase transcribes the LAC-I gene, it will result in a LAC-I mRNA. If RNA polymerase transcribes the Lac Operon, we'll have a polycistronic mRNA where all of the information to produce a LAC-Z, LAC-Y, and LAC-A protein products are on that mRNA. And so then we've got a single mRNA, and you'll see for each of the specific genes, we have our own start and stop codons. But again, it's all on this one mRNA. Then if the mRNAs are translated, we've got ribosomes now binding to the start codons and moving along, translating the mRNA and building the proteins encoded for by the mRNA. And so again, we see here that the LAC-I gene encodes the Lac repressor protein. And we see that these structural genes, LAC-Z encodes beta-galactosidase, an enzyme responsible for an early step in breaking down lactose. LAC-Y encodes a permease, which is involved in importing lactose into the bacterium. And then the LAC-A gene, which encodes a beta-galactosidase transacetylase, and actually it's still not well understood what the role of this is in lactose metabolism, but it's part of the operon. Okay, so we've got the structural genes, LAC-Z, LAC-Y, that encode enzymes responsible for breaking down lactose. These are on operon under the same regulatory control, so they have the same promoter and operator. We've got the LAC-I gene that is located elsewhere, not on the operon, but encodes the Lac repressor. Now let's take the situation where E. coli do not have lactose available to them, so in the absence of lactose. In the specific condition where lactose is absent, the Lac repressor protein will bind to the operator sequence of the Lac Operon. And so I'm showing that here. And hopefully what you can see is diagrammed here is that binding of the Lac repressor to this operator sequence inhibits, physically blocks RNA polymerase from transcribing the Lac Operon. So in the absence of lactose, the Lac repressor protein is bound to the operator sequence of the Lac Operon, and it inhibits and blocks transcription of the Lac structural genes. Now, when lactose is present, lactose is quickly converted into allolactose. And allolactose, or for simplicity we can just say lactose, is an allosteric inhibitor of the Lac repressor protein. What does that mean? So that means that lactose binds to the allosteric site of the Lac repressor protein, and binding of lactose to the Lac repressor protein inhibits the Lac repressor from binding to the operator sequence, or will, if the repressor is already bound, it will remove the repressor from the operator sequence. So now that the repressor is removed from the operator sequence because lactose, an allosteric inhibitor, is bound, RNA polymerase is now free to transcribe the Lac structural genes. So in the presence of lactose, we inhibit the Lac repressor and allow for expression, so transcription and translation of the Lac structural genes, so producing the enzymes that are necessary to metabolize lactose. And so hopefully you can see how the repressor here can act as an on-off switch of expression of the Lac Operon, where if the repressor is bound to the operator, you don't get transcription. If the repressor is not bound to the operator, you can have transcription. And then lactose itself, being an allosteric inhibitor of the repressor, is a way that the cell can then sense the environment and respond to the environment by changing gene expression. Okay, so that is the basics of the Lac Operon. What Jacob and Monod looked at was how mutations in different components of this operon can lead to a constitutive or a non-inducible operon. And so what do we mean by those terms? When we say constitutive, this would be an operon that is always on. So regardless of the presence or absence of lactose, a constitutive mutation would render the operon always on, meaning that the structural genes are being transcribed and translated. Conversely, a non-inducible system would be a mutation that renders the operon to always be off, so that transcription of the structural genes is not occurring. It's not inducible, meaning if you add lactose, that doesn't change whether transcription is then turned on. So it's non-inducible, means the operon is always off. And so to analyze different mutants, Jacob and Monod developed marrow-diploid Lac mutant strains by conjugation. And so what I'm showing you here is a very simple representation of an E. coli cell. And so this blue rectangle is meant to represent a copy of the Lac Operon on the bacterial chromosome. And also we'll remember that the Lac I gene is also on the bacterial chromosome, and that we've introduced in this marrow-diploid strain a second copy of the Lac Operon as well as the Lac I gene through a plasmid, specifically the fertility factor plasmid that we can introduce by conjugation. So we've created this marrow-diploid E. coli strain, a strain that has two copies of the Lac Operon and two copies of the Lac I gene, so essentially is diploid for those genes or for the operon. And by creating this marrow-diploid strain, we can then analyze different mutations to understand their function and understand more about regulation of the Lac Operon. Okay, so there's a lot going on this slide. What I want you to focus on first is at the top here, I am giving you a genotype of a marrow-diploid strain, where we have the genotype of one copy of the operon in the Lac I gene. We can say that's on the bacterial chromosome. And then the genotype of the second copy of the Lac I gene and operon in this E. coli. And so you can see that plus would mean wild type, so normal functioning. If you see minus, this would mean a null allele, so this gene is not going to produce functional enzyme. And you also see this C mutation in the operator sequence. And I've represented this genotype below. So here we have this genotype schematically represented, and here we have this genotype schematically represented. And this OC mutation is a constitutive operator mutation. So what that means, it's an operator sequence that the repressor protein cannot bind to. So since it's constitutive, since the repressor protein cannot bind, this operon with a constitutive operator mutation would always be on. So let's look at this now, whether the beta-galactosidase, the Lac-Z gene, and the Lac-Y gene are going to be expressed under different conditions. Specifically, are these genes going to be expressed when the operon is not induced, so in the absence of lactose, or are they going to be expressed in the presence of lactose under the induced situation? IPTG is a molecular mimic of allolactose, so sometimes that can be used to induce the operon. Okay, so we've got the Lac I gene. Both copies of this Lac I gene are going to lead to a functional Lac I repressor protein. And these repressor proteins are diffusible in the cell, and they can bind to any operator sequence. However, these repressor proteins, in the absence of lactose, are not going to be able to bind to the constitutive operator. So what does that mean in the absence of lactose? In the absence of lactose, the operon I'm showing you on the top will have the repressor protein bound to the operator sequence. And so RNA polymerase is not going to be able to transcribe, and so this one is going to be off. You are not going to have transcription. However, if we look at the operon on the bottom, we have a constitutive operator. And so this constitutive operator means that any repressor protein present is not going to be able to bind. And so in this case, RNA polymerase is going to be able to freely transcribe this copy, and so this will be on. And we can see here that we have a functional LacZ gene. So if this is on, we will have beta-galactosidase produced. But we can see this copy of the operon does not have a functional LacY gene, and so we are not going to have any functional permease produced in the absence of lactose. And so if we look at this, we can see that the constitutive operator mutation on one copy of the operon is not affecting regulation of the other copy of the operon. And so we would say the operators are cis-acting and that they're only affecting expression of the genes physically downstream of them in that same transcriptional unit. So cis-acting factors are typically regulatory DNA sequences that are only regulating the genes under their transcriptional control. Now, if we add lactose to the system, we'll remember that lactose is the allosteric inhibitor. So plus lactose, lactose is the allosteric inhibitor of the LacI repressor. And so in the presence of lactose, the Lac repressor from this operon will be removed from the operator sequence, and we will now get transcription of this operon, which is not producing functional Z, but is producing functional Y. So we'll have permease in the presence of lactose, and the operon on the bottom is constitutive. It's always on because of that constitutive operator mutation, so we'll also get beta-galactosidase. And so by analyzing this mirrodiploid strain, we can say operators are cis-acting. This constitutive operator is only affecting expression of the genes physically linked under its control. And we can look at whether the LacZ and LacY genes will be expressed under induced or non-induced conditions in this mirrodiploid strain. Okay, so here we have another mirrodiploid genotype where this genotype, this operon copy, is being represented here. This one's represented on the bottom. We've got our pluses and minuses. In this case, both operator sequences are normal, but we'll notice that on this copy, so right here, we have a non-functional LacI gene or null LacI gene. So this would mean that there is no repressor protein made from this allele. Again, this is all in the same cell. So this one, we do have functional repressor being made. And in the absence of lactose, this functional repressor will be able to diffuse in the cell and bind to any copy of the operator sequence. And so in this case, in the absence of lactose, we've got a repressor bound to both operator sequences, and transcription of both operons is off. So we are going to say in the absence of lactose, we have no beta-galactosidase produced and no permease produced. Now when we add lactose, when we induce the system, lactose acts as an allosteric inhibitor, and so again it will cause the repressor proteins to come off of the operator, turning transcription on. And so we have functional LacZ being produced from this copy, plus functional LacY being produced from this copy, plus. And so we can see here that even though we'll just say the bottom is the plasmid copy of the operon LacI gene, even though this is not encoding a functional LacI protein, the LacI protein expressed from the bacterial chromosome can diffuse in the cell and bind to any Lac Operon operator sequence. So we say that's trans-acting because it can affect expression of different molecules or genes encoded on different molecules in the cell. So that would be transacting, and transacting are usually diffusible proteins that affect expression of other genes. Okay, so I've gone through now a couple examples of analyzing different mutants. And so I'm giving you a table here where I'm listing different genotypes. Some are haploid, some are diploid or mirodiploid for the Lac Operon. And I'm asking you to say whether or not you'll get production of functional beta-galactosidase and permease under the uninduced or induced conditions. And so at this point, you should be able to now go through and analyze each of these different mutant genotypes to determine whether the Lac Operon will be expressed or not and whether it will result in functional protein. There's one additional mutation on here that I should explain. This IS is a super repressor, and it's a super repressor because it will not bind lactose. So this super repressor is still completely capable of binding to the operator sequence, but the allosteric site is mutant so that it will not bind lactose. And so you should be able to analyze that super repressor. Is it transacting or cis-acting? And is it going to result in a constitutive or non-inducible operon? And so I'm going to leave you with that and give you some practice at filling out this table on your own. And I will also end with saying that this is an example of negative regulation, where we have a repressor that is negatively regulating expression of the Lac Operon genes. I will say that the Lac Operon has another layer of complexity in there. It also is under positive regulation by catabolite repression, which I'm not going to get into in this video, but I should just say that regulation of the Lac Operon is more complex than what I'm presenting here, but I will save that for another video. Okay, I hope this was a helpful overview for you, and now you can go forth and analyze Lac Operon mutants to determine whether the structural genes are being expressed or not expressed.